Hey guys, so I just want to do a very quick overview of some meiosis stuff and compare it to some mitosis stuff. Okay, so looking at the start of my model, right, I have my full genome for this parent cell, but it hasn't been duplicated yet. Right? So this is like G0. The cell says, all right, time to do cell division. Let's go through the cell cycle, goes through G1, hits S phase, and now I have a duplicated copy of my DNA. It's very important to note that these duplicated copies are physically connected to one another. So the copy of 1A is connected to the original of 1A. They're linked. Okay? Same is true of all the chromosomes in this set. Understanding that's very important for understanding how the actual movement of these chromosome sets is working in both meiosis and mitosis. Okay? And so during metaphase 1 of meiosis, right, the chromosomes are lined up down the middle of the cell. Right? But it's very important that the homologous chromosomes are lined up together. Okay? So chromosome 1 is always going to be lined up with chromosome 1. Right? So I have 1A and 1B. Then I have 3B, 3B, 3A, 3A, 2B, 2B, 2A, 2A. Okay? Now, independent assortment tells us that these can be in any order, and that's why I have 1, 3, 2 rather than 1, 2, 3. Right? That's why I have A, B, B rather than A, A, A. Right? The order in which they can show up is completely random. That's what independent assortment is. Okay? The only thing that's important is that the ones always line up with the ones. The threes always line up with the threes. Okay? These are homologous chromosomes, and they must line up. Okay? It is at this point in time that crossover occurs between these inner homologous chromosomes, so 1A and 1B can touch a little bit and exchange information. In this model, I've denoted recombinant chromosomes with a green circle. So these are chromosomes that have gone through crossover and now have slightly altered allele combinations. Okay? So this is how they lined up in metaphase 1. Anaphase 1 is taking each half and separating them to the other sides of the cells. So both halves have 1, 2, and 3, okay? But whether they have A or B is now arbitrarily assigned through independent assortment. Okay? We then separate them with telophase 1 into 2 new sets, right? And so now I'm working with this set here and this set here. They're now separate things, okay, at the end of PMAT1. What PMAT2 is doing is dividing the sister chromatids apart and moving them into opposite directions. Okay? So 1A and 1A are now going to get ripped apart. Okay, that connection that was holding them together is being ripped, and they're getting pulled to opposite ends. Same is happening with this side here. Okay? And so here, this left side becomes this gamete. You can see that it has one copy of 1, one copy of 2, and one copy of 3. This right side becomes this gamete. It has one copy of 1, one copy of 2, one copy of 3. This left side makes this gamete, one copy of 1, one copy of 2, one copy of 3. And this right side makes this gamete, one copy of 1, one copy of 2, and one copy of 3. And so this is what our gamete outcome looks like, right? They all have one copy of each chromosome. That's necessary in order for a baby to be made, right? The baby needs all the chromosomes. Like I can't have 2 2s here and 1 1. I need 1 2 3. Okay. So again, that's represented here in this overview where I have my duplicated sets. They get split through PMAT1 into 2 sets, and they get split again into 2 sets. And these are our 4 gametes, each with one of our copies. If we're looking at mitosis, right, the main difference here is the way in which our metaphase lines up our chromosomes. In mitosis, the homologous chromosomes don't need to be paired. Instead of these 2 parallel columns, there's just this one column. And it's arbitrary what order they show up in. It doesn't matter, right? So here I have 1A, 2B, 1B, 3A, 3B, whatever order is in, doesn't matter. Okay? Because anaphase is just pulling the sister chromatids apart, right? Pulls it apart, and then both daughter cells are going to end up with a full copy of the DNA, right? So they have 2 copies of 1, They have 2 copies of 2. If I had done this right, I forgot to put 2A in here. 2A. Dummy Mr. Lavadier. All right, so here's 2A. And 2B. 2A, 2A. Cool. All right, so they have 1, 1, 2, 2, 3, 3. It has all the DNA. And so this way in which the metaphase versus metaphase 1 lines up the chromosomes is very important for the differences in the outcomes of mitosis versus meiosis. So here's our meiosis metaphase 1, right? You can see homologous chromosomes are next to each other, but in mitosis, they just lined up in one row. Cool. That's it. Bye.